Great.
Start.
All right.
You want to kick us off?
All right. So good morning, everyone. Thank you so much for joining this pre-meeting. This was really just a testament to how passionate everyone here is about kidney cancer, but also about the rarer variants of it. So I think we wanted to spend a good amount of time this year on some dedicated sessions with 4 experts in the field. Rana, do you want to start off? We had a pre-meeting survey.
So thank you all for humoring us with our pre-meeting survey. We know it went out on Monday, and we basically gave you 3 days to complete 20 questions, so we appreciate that. But we thought this would be interesting to kind of just level set where people are at. So this is us, you know us. All right, here are a lot of slides. Okay, so we're going to start through. So we asked a couple of questions. You can kind of see them here. Are you primarily clinical, non-investigator, or both? And we have a pretty good mix of mainly clinical, but many people do both clinical and translational work, and then some non-clinical people here in the room, so welcome. What's your medical specialty? I think we've got good representation, heavy medical oncology, but we've got good representation of urology, pathology, and other that's radiation oncology there. Opportunities for next year for people who completed the survey. All right, so then we went through just a series of questions about frontline treatment for papillary, for all of the variant histologies, and then a little bit of questions about clinical trial design and where there's gaps in the field. And when we asked the question, what's your frontline treatment for papillary RCC, resoundingly, the bulk of individuals selected IO-TKI, and you can see the IO-IO TKI mono there. For Chromophobe, same thing. I will highlight that in the other category, it largely was lenvatinib, everolimus, or base treatments. Okay. For translocation RCC, also exceedingly IO-TKI, and HLRCC, we also added bev or lorlatinib, and actually the bulk of the field is choosing that as a frontline. And that one other regimen there is actually bev or lorlatinib atezo. Currently with the phase 2 that that RAM's leading from the NCI. And then with regards to medullary, you can see that there's really no IO-based treatments at all that are selected for this disease. Everybody's largely using frontline chemo. And then now we've kind of shifted gears, so frontline, the next set of questions are really around adjuvant therapy. Have you given adjuvant pembro to a patient with variant histology RCC? The bulk of individuals have never given pembro to variant histology. Some, yes, but very, very rarely. 5, yes, sometimes nobody, yes, always. So still a gap in the field what to do for these individuals adjuvantly. Do you think we need a dedicated adjuvant trial? And the bulk of individuals actually answered, yes, we do, and it needs to be tumor specific as opposed to just lumping everybody into one category. And then we asked the question about terminology. This comes up a lot. How do we think about the variant histologies and label them? And the bulk of individuals answered, name the specific subtype, stop grouping them, they have different biologies. And then what's the appropriate control arm for frontline IO-TKI trials in variant histology RCC? This is kind of a poorly worded question. We should have just said frontline trials as opposed to frontline IO-TKI. But the bulk of individuals selected cabozantinib. What's in that 8 other was actually IO-TKI. And then what's the biggest challenge in conducting clinical trials for variant histologies? For this specific question, we forced people to pick one answer only. And the main challenge is patient recruitment and rarity. And so I think this really speaks to the need for us to come together and collaborate, because the only way for us to move the field forward is actually by doing that. And you can see some of the others, the heterogeneity of the subtypes was another one that showed up, and limited understanding of disease biology. And then some questions about, do you routinely order somatic tumor profiling for variant histologies? Most individuals in some fashion are doing this, whether it's for all patients or select patients. And then with regards to the germline testing, not as common for everybody, but for select patients considering doing this. And then for this questions, where do you consider the most important gaps? And this was a check all that applies, so there's people could have answered, could have checked every box or could have checked one box or no boxes. Actually, I think you were forced to check a box, so if that's where the abstains come in. But I think the biggest items here were actually identifying therapeutic targets and better understanding of disease biology. And then which variant histology subtype would be the highest priority for therapeutic development? You can see it's hard to pick one, papillary, but then close second, chromophobe, translocation. So I think we really need to be thinking about dedicated trials across each of these specific disease entities and come together as a field, certainly within the pediatric community and other communities, lung cancer community, they have been able to conduct trials in very, very niche populations, and I don't think that our community can't do the same. And then similar question, if you had to prioritize conducting a clinical trial in one variant histology, which one would it be? Papillary and actually translocation were the top two on the list here. And then if you had to pick one disease setting, neoadjuvant, adjuvant, first line, later line, the bulk of individuals selected, we need a metastatic frontline trial across the distinct variant histologies. And then what would be most helpful to advance research for variant histology, RCC? Resoundingly, the most commonly selected answer, 51 individuals, across everybody that completed the survey, was multi-institutional consortiums. So I think we really need to take this to heart. I think this is the group that is going to help set up such consortiums. And so if this is the way that we're going to be doing clinical trials and recruiting patients and collecting specimens, I think we really need to come together and start to convene around that. And I'd love to hear sort of some of the efforts that Srini and others are kind of beginning to roll out about doing this very thing in a collaborative manner. And then the second was patient registries, which kind of links to the multi-institutional consortiums. And then the last question was, how optimistic are you about the future of variant histology RCC? This is probably more a question about people's personality and style than it is about the actual field. Only 9 were super very optimistic, but moderately optimistic, the bulk of the people in the room. There were some cautious optimists, 2 pessimists that, we've got to, whoever you are out there, we're going to find you and convert you over. And with that, we will start. So,
all right, so we have Dr. McGregor here from Dana-Farber, who's going to lead us off.
All right, so I'm the guinea pig if you have the podium and the microphone works here.
Yeah, and now there's a lot of handling. They've got to hold the mic, they've got to do the clicker team, so we may want to think about that because the speakers are having to hold 2 things, like the mic and the clicker.
Yeah. Anyway, well, thanks so much for the opportunity to be here. I'm excited to be kicking off this session, and we'll get going. So I was tasked to talk about updates in papillary renal cell carcinoma, and I think one of the key take-home points, hit the button accidentally, is that when we think about papillary RCC, this is something that's a histologic diagnosis, is not a genomic diagnosis. And just how we sort of take in non-clear cell and said, really, this is not just non-clear cell, this is a bunch of different divergent histologies or individual histologies. I think we're going to get the same thing with papillary RCC, that we've been lumping all of these diseases into one disease, but really it's a lot of different diseases. We have, papillary RCC may be a true MET-driven RCC, but collecting duct sometimes like papillary, tubulocystic, FH deficient, out-translocated. In each one of these, we treat incredibly different. Now we know about biphasic hyalinizing simomatous RCC, which has a very aggressive course. So at the end of the day, I think the key thing we have to think about as investigators is this is a heterogeneous group of tumors with overlapping morphologic findings, but very distinct genetic molecular alterations and different biologies that we have to target. So as we start looking at papillary RCC, I think we start appreciating this. And I think we look no further than PatMed. This is an amazing trial that really showed that we can do these trials in these rare histologies conducted by Monty Paul, looking at histologically confirmed papillary RCC. And they had 4 arms looking at sunitinib, cabo, and then the idea that papillary RCC is MET-driven, there were arms with crizotinib and savolitinib. And then very early on, those 2 arms were closed for futility, again highlighting that papillary RCC was very distinct, a very heterogeneous group of patients, and so a lot of these diseases were not MET-driven. But what we did see is while there was no OS benefit with the updated analysis just published, there was an improvement in progression-free survival. And so this was one of the first trials that sort of advanced the field forward in papillary RCC and established cabozantinib as a standard care, which is why I think everyone selected that as the appropriate control arm for frontline trials. But we can certainly do better. And so PapMet, amazing effort. We try to take this histologic subtype, but we now appreciate that's not probably going to be the best approach. And so Savoir was a genomic-driven approach and said, let's not just look at papillary. Let's say we want to look at those patients who have a MET-driven disease. And while this trial was closed early, there was some encouraging signal that we saw an improvement in progression-free survival, 7 versus 5.6 months. And more importantly, it could be done. So this was a large international effort, but it was successful and they were able to accrue patients to this trial. When you add durvalumab, we had a 57% objective response rate. And so we await SUMAIDA, which has now completed accrual. Again, looking at the role of savolitinib and durvalumab versus sunitinib versus durvalumab monotherapy in those patients with MET-driven RCC. So again, highlighting that this is a subtype of papillary renal cell carcinoma. And as everyone already heard, beverlatin, we finally saw the data published in New England Journal, so congratulations to the team at the NCI. I think this, to me, was amazing data. It sort of changed the way I think about this in the clinic. And again, highlighting that these 2 different cancers, FH deficient and all the other papillaries, all grouped the same, but if you look at the breakdown, FH deficient RCC, really impressive, over 70% objective response rate with a combination of beverlatinib versus 35% with sporadic papillary RCC, duration response approaching 2 years. So really, really exciting data. I think it's an amazing option for our patients, but again, highlighting that all these diseases are very distinct. What about other options that we can think about in this setting? I think we have lenvatinib, everolimus. This was, published quite some time ago. Only 20 patients, relatively disappointing objective response rates at 15%. So I think as we think about options we think about for papillary RCC, I don't think Len-Ev is going to be jumping to the front of the pack. But what about immunotherapy? So as we look at the first experience with immune checkpoint blockade in the divergent histologies, looking at pembrolizumab, papillary was certainly the one where we saw some of the most exciting activity overall, with a mean duration response of 29 months. And so this was sort of the foundation, said, hey, we can start thinking about immune checkpoint blockade in those patients with divergent histologies. But I think no one would be using monotherapy checkpoint blockade. We've had the results published from Sunny Forecast, which was looking at the role of Nivo-Ipi versus basically investigator's choice. So a lot of these were sunitinib, and then over time they amended to include other options. But what we see overall, looking at those papillary, again, this very heterogeneous group of tumors, we had a close to 30% objective response rate versus 20% with standard of care, but over a 30% PD as best response rate. And so if we look at even the retrospective data, look at the Nivo-Ipi, it's not quite as good as what we see with clear cell, but there certainly is some activity. And akin to what we saw with atezo-bev and some other data, there seems to be a role for PD-L1, and those who are PD-L1 positive maybe have the greater benefit, but again, these are small numbers. But this data does support the role of Nivo-Ipi in select patients with papillary RCC. And as we already heard, IO-TKI is sort of everyone's go-to frontline standard, and that's based on 2 major trials. This data from Joe Lee looking at the combination of cabo-nivo. There's the 2 cohorts, as everyone was aware, the Chromophobe, no response to shrinkage, so it's closed. But when we look at cabo-nivo in the papillary, we had close to 50% objective response rate. And again, if you look at this and you try to tease out a little bit further, if we start looking at the genomics and we appreciate there are these different types of tumors, those FH mutated disease, they actually had a really impressive response rate, 80%. So again, highlighting that these tumors are a little bit different. NFS2 also a very high response rate. So just highlighting that papillary is a histologic finding, not a genomic finding, and we need to probably thinking about this at a different level. And then we have a very large trial, close to 160 patients with divergent histology, looking at the combination of lenvatinib and pembrolizumab. Again, very similar numbers to what we saw with the data for Cabo-Nivo, with a 54% objective response rate. And akin to what we see with IO-TKIs in clear cell, lower PD rates as best response. So again, similar to how we go in the clear cells, we're thinking, what are we going to do in the clinic? I think if we have a patient where they have a pending visceral crisis, an IO-TKI offers the best data. I apologize, I don't know what happened here. Oh, this is looking, I'm sorry, the animations got all messed up, but I do want to highlight that you need pathology review, you need expert pathology. And this trial, which is like a large international trial, if you just look at the appendix and you say, well, what did people think it was when they went in? What did people think it was at the center review? There was a difference. So patients who were initially classified as papillary may have been classified as unclassified or translocation or other. And so it just highlights the need that we start seeing these divergent histologies, especially as we're getting more and more subtypes, engaging with your pathologist, and as we heard, getting some of those the genomics to help really better classify what is going on. And this is just highlighting sort of the different combinations that are out there overall when we start looking at the different subtypes, and just highlighting that when we think about the current disease landscape, the IO-TKIs certainly have the best efficacy, albeit in single-arm phase 2 trials, but other IO combinations certainly have a role. And the real question is, which approach is best, right? And so I think we're very excited about the single-arm data. If you look at some of these retrospective analysis and you try to say, do we really need to IO-TKI? Can we do TKI alone? If you look at this overall, there doesn't seem to be a clear benefit in these analysis from TKI monotherapy, from IO-TKI, but again, these analyses are always confounded by a lot of factors, and we really need to support ongoing trials. And to that extent, we have Stellar 304, which is looking at the combination of zanzalintinib with nivolumab versus sunitinib, randomizing 2-to-1. And then equally important is the PAPMET-2, which is run through the cooperative groups by Ben Mon, looking at the combination of Cabo with Atezo versus Cabo. And I think this trial is really important because it's the same TKI in both arms, and you're looking, does the addition of IO truly make a difference in the front line? And so I think, to the point of where are we right now, should we be pessimistic, optimistic? I am optimistic because I think we've learned a ton, and we're starting to take lessons from what we've done and do a better job. And I think the trials that are ongoing are going to be critical, and I think we can do better. We can work to find the right combinations, the right control arms, and work across institutions to do this. Because ultimately, this is a heterogeneous group of diseases, and target therapy, immunotherapy play a critical role, but which target therapy, which immunotherapy, for which type of papillary is going to be critically important as we continue these studies.
Thank you, Ben.
Great.
Thank you. All right, we're going to go on to our next speaker. We're going to do questions at the end. Lisa Henske from Brigham and Women's. Thank you.
All right, is the speaker working? Great. Super happy to be here today. Let's see if I can advance. So I wanted to start by acknowledging the amazing, amazing people that have helped with the work that I'll be talking about today, especially people who are here today, who include Tony, Vincent Zhu, David Braun, and Carmen Priolo, and the members of my lab team, many of whom are also here today. So, take-home message, a lot is happening in Chromophobe right now. There are encouraging immunotherapy results, some of which Brad just mentioned. We have new single-cell RNA sequencing data that was just published, thanks to David Braun and Chris Levacki and others. And we have new targets, ferroptosis, and very importantly, KIT, with a antibody-drug conjugate trial going on right now. So just to remind you about the very unusual hallmarks of chromophobe kidney cancer, they include these whole chromosome losses, very mysterious, very few driver mutations. So P53 is the most common in 32%, but many tumors don't actually have an identified oncogenic driver. The highest mitochondrial DNA mutation rate in the TCGA, very, very high glutathione levels. This was discovered by Carmen Priolo, who's here today, and median survival in the metastatic setting of 20 months. And we think there might be two different pathways that are driving the development of chromophobe. One involves mitochondrial DNA mutations that may be exacerbated by oxidative stress and account for these high levels of glutathione, and then secondly, nuclear mutations that include the mTOR P10 pathway, which is how I actually originally got interested in chromophobe, because these tumors occur in individuals who have tuberous sclerosis and Birt-Hogg-de-Bay syndrome. So I'm just going to briefly mention the single-cell data set. This was 3 chromophobes, 2 low-grade oncocytic tumors, 1 renal oncocytoma, and matched normal kidney. This was a really amazing effort by a lot of fantastic people, and there's a lot of data there that we hope you will mine, but the hits include NRF2 signaling, and I was happy to see that ferroptosis also arose from that analysis. We also, not surprisingly, saw that there was reduced immune infiltration and lower levels of immune checkpoint inhibitors than clear cell kidney cancer or papillary kidney cancer. So Brad already mentioned some of these trials. These are the trials from prior to 2023, but we now have 2 trials that are showing potential responses to immunotherapy in a subset of individuals with chromophobe, including the Keynote B61 trial and the Sunny Forecast trial. And we really would like to better understand why did these particular patients respond, of course, and that's something that I don't know the best way to get to those answers, but I'm optimistic that we can do that. So I think the one limitation right now in what we know about chromophobe is that most of the data comes from the primary tumors, not the metastatic sites, and we really need to understand better the immune tumor microenvironment of the metastases. So we have been looking at ferroptosis for quite some time. That started because we found these super high levels of glutathione in the tumors. And we have previously shown that if we limit cysteine uptake, which is, of course, the rate-limiting step for the synthesis of glutathione, we can impact tumor biology in the models we have of chromophobe. And this is just one example from prior work showing that if we treat with IKE, which blocks cysteine uptake, we can decrease tumor growth in mice. But in unpublished work, we've been looking at another arm of the ferroptosis pathway, and this involves CoQ10, also called ubiquinone. So there's a protein called FSP1, or ferroptosis suppressor protein 1, that is responsible for the reduction of ubiquinone, and that allows this other arm of the ferroptosis pathway to be active, where actually the reduced CoQ10 is able to block the oxidation of membrane lipids. So in work that's not yet published by Samer Salem in my group and others, working with Carmen Priolo and others, we have looked very carefully at FSP1, or ferroptosis suppressor protein 1. You can see here that it's highly expressed in chromophobe in the TCGA, the second highest level, and it does seem to correlate with the survival in chromophobe. So if you have a high FSP1 level, that might suggest that those tumor cells are more able to resist ferroptotic death, and patient survival is therefore less. So Samer found that if you inhibit both FSP1 and cysteine uptake, SLC7A11, using IKE, you can have a synergistic effect on the survival of chromophobe cells. So here we're basically working on both arms of this ferroptosis equation. So this chart shows the many different doses of an inhibitor of FSP1 called IC-FSP1 combined with IKE. And if you look at the colored squares, you can see that there's very little effect in the cells in vitro of inhibiting FSP1, and at this dose that's highlighted here, very little effect of inhibiting cysteine uptake, but the combination has pretty clear synergy. But the greatest result so far has been that in vivo, if we inhibit only FSP1, in this case using shRNA, we can quite strikingly reduce the growth of chromophobe-derived tumor cells. You can see here the actual tumors and the growth kinetics of 2 different shRNAs for FSP1. So it turns out that there's quite an interest in developing FSP1 inhibitors, still at a preclinical phase, but this might be a future direction that could lead to therapeutic opportunities for Chromophobe. I want to end by talking about what might be the most immediately relevant aspect of Chromophobe right now, which is KIT. So, KIT has the highest expression in Chromophobe of any tumor in the TCGA. And this has been known for a long time that KIT is highly expressed because it's used as a diagnostic marker of Chromophobe. KIT, the ligand for KIT, which is stem cell factor, is also highly expressed in Chromophobe. You can see it's the second highest in the TCGA. And in work with Michelle Al-Shaweri and Michelle Hirsch and others, we've found that the majority of metastatic chromophobes at our site express KIT by immunohistochemistry. And in other data that we've just learned about, it looks like KIT is expressed at the RNA level in almost all metastatic chromophobes. But it's wild type KIT. It's not mutant KIT like in GI stromal tumors. So that might explain why chromophobes have not conventionally responded to inhibitors of KIT, and sunitinib, among others, is an inhibitor of mutant KIT. And we don't actually expect a response to drugs like imatinib that inhibit the mutant form of KIT. But we do think there's a lot of optimism for antibody-drug conjugates targeting KIT. And we've been working on this to develop novel antibody-drug conjugates with Bill Sellers at the Broad. You can see here that 2 chromophobe-derived cell lines are having a response in the orange and the green to a KIT antibody-drug conjugate, and clear cell carcinoma, which doesn't have high KIT expression in these in vitro studies, is not responding. So we're working on this, but in the meantime, just the greatest thing has happened, which is that a company based in Korea called Novelty Nobility has a phase 1 trial of KIT antibody-drug conjugates going on right now. It started in 2025, and it's just wonderful because the inclusion criteria include Chromophobe. So we're really hoping that we'll have some enrollment and see a signal on this trial, and I'm hoping that everyone in the room who has individuals with Chromophobe in their practice will consider this trial. It's open at 7 sites in the US, at MD Anderson, Ohio State, Michigan, and Case. So we really need to get Chromophobe enrolled. As far as I know, there have been no Chromophobe tumors enrolled yet. It's been mostly enrolling small cell lung cancer so far. So to sum up, a lot is happening in Chromophobe. It's really it's wonderful to see. The whole community, I think, is galvanizing around what progress we could make in Chromophobe, including some encouraging immunotherapy results. We need to know more about why those particular individuals responded. The possibility that we could target ferroptosis, although that's a bit in the future because the drugs are not yet available, and KIT, which we could be learning more about right now through this trial that's open. Thank you guys so much.
Yeah. Thank you, Lisa. Thank you, Lisa. Next up, we have Srini, also another Boston representative here, and a passionate investigator in translocation RCC.
Okay, great. Thank you very much. So I'm going to talk a little bit about some emerging targets in translocation RCC. So just by way of background, I think most of this audience knows translocation RCC is a genomically defined subset of kidney cancer. And in that way, it's a little bit distinct from papillary, as Brad was mentioning, which is somewhat heterogeneous since it's more of a histologic diagnosis. So in this case, all translocation RCCs have this defining lesion, which is a fusion of an MITF transcription factor, most often TFE3, and that generates an oncogenic transcription factor that is constitutively nuclear and constitutively active and drives the cancer, as shown on the right, and can be easily detected by FISH or other NGS assays. So several years ago, we started to get interested in beginning to define the genomic landscape of TRCC apart from that driver fusion. And what's remarkable about it is that it's quite bland, as you can see from the empty commute plot on the left side, which is that in most cases, there are few or sometimes no other alterations apart from the driver fusion. So it's really this TFE3 gene fusion that drives the cancer. And if you look at the copy number profile as shown on the right, sometimes there are copy number alterations, but sometimes there's nothing. And so it's really this solitary fusion that defines the cancer biologically. And so that raises the question as to what are actually the targets that we can go after in this genomically defined subset of RCC. And to identify new targets, efforts like the cancer dependency map, which perform genome-wide CRISPR screening across an array of different cancer cell lines, have been extremely useful. There have been about 1,000 different cell lines of many different cancer types that have screened via that effort, but translocation RCC is not included. So recently, we performed genome-scale genetic screening in 3 TRCC cell lines and calibrated the data against the data in the cancer dependency map. And just to orient you to what's shown in that scatter plot on the right side, the x-axis shows the strength of a gene's dependency. Each dot is a gene. So more to the left means that if you knock out that gene, it's more of a dependency in that cell line. And the y-axis further down means that it's more of a selective dependency in TRCC versus other cancer types. So that lower left quadrant in the pink are the dots. Those are the genes that represent selective dependencies in TRCC. And you can see that the most outlier there is TFE3, as well as the TFE3 fusion partner, ASPSCR1, which really shows that the fusion is the top selective dependency in TRCC versus other types of cancer. But there are also a bunch of other dots in that left lower quadrant because the fusion itself, for various reasons, is difficult to target directly. And so if you do pathway analysis of those dots in the left lower quadrant to see what pathways are more selectively essential in TRCC compared to other subtypes of kidney cancer, the top pathway that's enriched amongst TRCCs is OXPHOS, or oxidative phosphorylation. So oxidative phosphorylation refers to the pathway whereby ATP is generated from energy in the bonds of glucose via the mitochondria with oxygen as the terminal acceptor. And despite the fact that this is the most efficient way to generate ATP, many cancers, and especially clear cell RCC, do what's called the Warburg effect, or aerobic glycolysis, where they shunt pyruvate to lactate even in the presence of a sufficient oxygen tension. So what this suggests is that TRCC is metabolically the opposite of clear cell. It's actually metabolically divergent from what we see in clear cell, which is that it has this reliance on oxidative phosphorylation. And you can see all the way on the far right all the many genes that were pulled out in this unbiased genetic analysis that are involved in oxidative phosphorylation in the electron transport chain and are essential to TRCC cells. So that pathway appears to be essential to TRCC cells, but I also mentioned that the main genetic alteration in TRCC is this TFE3 fusion. And so that raises the question of whether it's the fusion itself that's metabolically rewiring TRCC cells to rely on oxidative phosphorylation. So we performed ChIP-seq against the TFE3 fusion in a bunch of different TRCC cell lines. And you can see that if you look at the overlap amongst all those different cell lines, and see what genes the TFE3 fusion is binding next to, oxidative phosphorylation-related genes are highly enriched amongst TFE3 fusion targets. On the right side, you see the citric acid cycle in the electron transport chain and all of the genes that are involved in those pathways. And each of the orange boxes represents a gene that's a direct transcriptional target of the TFE3 fusion. So you can see that the fusion is really coherently activating flux through oxidative phosphorylation in the citric acid cycle. And that may explain why TRCC is uniquely dependent on this pathway. And on the bottom left, you see IHC showing how highly expressed some mitochondrial complex 1 components are in TRCC on the top versus clear cell on the bottom. So there are actually complex 1 inhibitors that have been tested clinically, but they have a very narrow therapeutic window because it's obviously an essential process. And so we wanted to see whether there were other ways that we could impinge on this same biology that may perhaps be better tolerated and more selective. So we went back to our CRISPR screening data and looked at selective metabolic dependencies in TRCC versus clear cell. And the TRCC selective dependencies are the orange dots on the left side. And you can see that the top selective dependency in that analysis for TRCC is this gene called eglin-1. Eglin is a prolyl hydroxylase that, in the presence of oxygen, hydroxylates HIF-1 alpha and targets it for degradation. HIF-1 alpha, when it's stabilized, promotes the glycolytic reprogramming of the cells, so away from OXPHOS and towards glycolysis. So you can imagine, based on what I said, why knocking out eglin-1 or inhibiting its activity might be a selective dependency in TRCC, because if you inhibit the activity of eglin-1, you would stabilize HIF-1 alpha, reprogram away from OXPHOS and towards glycolysis, and in this very OXPHOS-dependent cancer type, that could represent a selective vulnerability. And indeed, that's what you see, which is that when you knock out eglin-1 in TRCC cells, it's a vulnerability on the top right, whereas clear cell RCC tolerates this fine. And when you knock out eglin-1 or inhibit it using a pharmacological inhibitor that's actually approved for the treatment of renal disease-associated anemia, you can see that it reduces the oxidative phosphorylation capacity on the bottom right there and is selectively essential in TRCC cells. So I'll also talk about a second potential target that came out of this CRISPR screening analysis. So I talked about OXPHOS, which is the top enriched pathway that's selective for TRCC. The second top enriched pathway is the E2F pathway, which is downstream of CDK4 and CDK6. And I'll just go briefly over this, and additional details are in the poster by Shikha Gupta here. So the CDK4/6 pathway, CDK4/6 complex with D-type cyclins, and are responsible for phosphorylating RB. When RB is phosphorylated, it releases E2F, which then promotes G1S cell cycle progression. So these inhibitors against CDK4/6 are obviously approved in breast cancer, where they've been very effective in combination with ER antagonists. And the idea there is that in order to really potently inhibit this pathway, you need to both inhibit CDK4/6 kinase activity as well as the complex D-type cyclin. And in breast cancer, ER is known to activate cyclin D. So the analogy here in translocation is we believe that the TFE3 fusion is directly activating cyclin D, as you see from the ChIP-seq tracks on the right. And it also indirectly activates cyclin D by activating mTORC1 signaling, which enhances cyclin D1 translation. And so the idea here is that maybe this is an analogy to breast cancer, where CDK4/6 is activated by the fusion and the D-type cyclins are as well. So to try to investigate this further, we first looked at inhibiting just CDK4/6 alone. And you can see that when you knock out either CDK4 or CDK6, TRCC cell lines are highly dependent on that, either one or both. That's shown on the left side, where further down is more dependent. When you treat with a CDK4/6 inhibitor, here this was palbociclib, there is a suppression of proliferation that's very potent, but as soon as you wash out the drug, the cells regrow, so it's cytostatic but not cytotoxic, and that's similar to what's seen in other cancer types. And so really, these agents are best used in combination. And so based on what I told you in terms of the pathway, we were trying to figure out what a potential combination agent would be with CDK4/6. And TFE3 fusions have been known in multiple studies to activate mTORC1 signaling. And as I mentioned, they also directly activate cyclin D, and the mTORC1 also activates cyclin D. And so we turned to this mTORC1 selective inhibitor, RMC-5552, which is currently in phase 1. So this is different than the other mTOR inhibitors that we're familiar with in kidney cancer, in that it's extremely selective for mTORC1 versus 2 due to its mechanism of action. And unlike other mTOR inhibitors, is able to result in complete 4-EBP1 inhibition. So you can see on the left side in that Western blot, when you treat with increasing doses of RMC-5552, you see almost complete ablation of phospho-4-EBP1, and that results in a potent downregulation of cyclin D1 protein levels, which is shown on the bottom left. So this is something that we don't typically see with other mTOR inhibitors, and indeed when you combine RMC-5552 and palbociclib, you see synergy and now an increase in apoptosis with the combination, suggesting that that cytostatic effect is being converted to cytotoxicity. In an in vivo model, and this is the fastest growing cell line xenograft we have of TRCC, the combination does lead to improved survival and a delay in tumor outgrowth as well as an increase in apoptotic markers, whereas the single agents do not. So this is one potential combination that we think could have potential in TRCC to be developed clinically. So I think to conclude, we really need effective systemic therapies for TRCC that are directly tied to the mechanism of this cancer versus other subtypes of kidney cancer, as opposed to repurposing clear cell therapies for TRCC. I think some of the data I showed and other efforts are starting to better define the molecular landscape and potential vulnerabilities in TRCC, and CDK4/6 and eglin-1 inhibition are two potential therapeutic targets, and there may be others in the pipeline as these types of studies go forward. And finally, I'd just like to draw attention to a recent effort in partnership with Count Me In at the Broad Institute with Elizabeth Mullen as a partner on the pediatric side. We've recently launched this TRCC project, which is a direct-to-patient, patient-partnered research project where patients can go to this website and sign up to participate, to submit their clinical samples, data, and other information, so we can build a larger cohort of both adult and pediatric TRCC and start to understand the clinical features, the patient pathway to diagnosis, the patient journey, as well as molecular features based on genomic profiling of that cohort. And I think this will help to build the first generation map, molecular map of TRCC that will allow for further and more robust target nomination in future studies. So I'd like to close by thanking all of the members of the lab, including the postdoctoral fellows whose work I presented today, Zhao Li and Shika Gupta, collaborators, Tony, Sabina, and many others shown here, and funding sources. Thank you.
Amazing.
Amazing. Thank you for that presentation. And our last speaker is Yingbei Chen from MSK. She'll be discussing unclassified RCC.
Good morning. Thanks for the opportunity to speak today. Okay, so I'm left with the unclassified variant. So before we define what is unclassified, we talk about the current classification system. So in the most recent 2022 fifth edition of WHO classification, there's sort of major structure change. So on the left-hand side, you see we maintain the more histologic defined type of renal cancer, but we also added the more molecular defined entity. These are mostly in the last 2 decades identified from the previously histologic subtypes, and some of that has been discussed today. So the unclassified is mainly, in the high grade. We're mainly focused on the high grade group, which is currently has also a name of not others, NOS or not other specified. So which is a little bit confusing, but that's the current classification. And aside from the high grade, you can see in the oncocytic group, we also have some NOS oncocytic tumors, which usually refer to those very low grade pink cell tumors. We're not exactly sure what they are. So in the unclassified group, so mainly they're representing either the unknown or the gray zone areas of classification. So from a morphological perspective, so there are usually tumors with papillary features, tumor with high-grade poorly differentiated morphology, or tumor with oncocytic eosinophilic features. So among today's topic, the papillary RCC, the high-grade spectrum. With molecular analysis, now we have identified all these distinct molecular-driven subtypes, and whatever remain that we are not sure become unclassified. And interestingly, many of the molecular defined entity, but particularly the FH deficient RCC, also even the translocation one can have a broad morphological spectrum. So they can be sort of closely mimic a high-grade papillary, but also can mimic the traditional, more poorly differentiated, even like collecting duct morphology. For the oncocytic eosinophilic group, one major mimicker is Chromophobe RCC, and in recent years, there are also more defined, like eosinophilic solid, cystic RCC, and some other mainly mTOR-driven type of pink cell tumors. So, particular for papillary RCC from the TCGA studies, you notice that one major things for the classical PRCC is the copy number, chromosomal level copy number changes, as shown on the left, upper left. You can see that's one of the key features to distinguish PRCC group from other type of RCC. However, if you look at higher resolution in the lower left, you can see there are at least two subgroups of PRCC that mainly so-called, they used to be type 2 that has quite distinct chromosomal alteration numbers. Some of those actually closely resemble some of the group of unclassified RCC, and also include those other molecular identified entity listed on the lower left, lower right. So in the current classification, thus far, so papillary rely on exclusion. So we emphasize it doesn't have any specific features of other renal cell carcinomas with papillary just growth pattern. And also, as we mentioned before, the type 2 no longer was recommended because the heterogeneous features of them, but still remain this gray zone that in the high-grade papillary RCC, when you have a complex architecture that's beyond the typically well-circumscribed papillary growth pattern, and with infiltrating growth, so then that means also mimicking like collecting duct carcinoma. And they also like the defining molecular features of any known established subtype. So should we call it the high-grade papillary, or should we call it unclassified? And similar thing for collecting duct carcinoma, because this is one of the types has not been defined molecularly. So for clinical diagnosis, we emphasize on the location. It's mainly a tubular growth pattern, infiltrative, and again, it's rely on exclusion. So here, collecting duct become, for this growth pattern, collecting duct become important, distinguished from unclassified, and we don't really have a very good handle on that. But nonetheless, for most of the molecular defined entities, in recent years, we have developed very powerful ancillary tests. So here, for example, when you have a renal mass biopsy, first we decide whether it's primary or not, and whether it's renal cell carcinoma versus the upper tract urothelial cancer. And then even for a small biopsy, we will be able to utilize some of those ancillary tests in combination, for different pro and cons, for either whether it's immunohistochemistry versus FISH versus RNA-based fusion assay versus other clinical correlation. We can actually pretty nicely define most of the molecular defined entities nowadays. So the remaining, group of cases that usually pose a challenge for biopsy is, high-grade clear cell totally lost the clear cell the typical clear cell morphology. And the CA9, although is possibly over 90% of clear cell RCC can be lost, even PAX8 can be lost. So those tumors, unless we do sequencing, it's really difficult to know what exactly what type that is. And then again, the papillary and unclassified, because they are less well-defined and we don't have specific IHC test, those also would be rely on more molecular analysis. And another thing is limited material on biopsy often render them cannot be classified because either we don't have the assay or don't have enough material to do the assay, so those are not really unclassified per se. So the molecular analysis in unclassified here is the one study we conducted actually quite a number of years ago now from the 62 cases of primary resection cases. Here, the one important message come out is a large subset of them harbors NF2 mutations. And in another subset that come out is the mTOR complex, one hyperactive subgroup, which is the yellow group here. So this two group has very distinct clinical behavior at that time with those treatment options available then. And you can see FH at that time was still part of this group and has worse outcome. So for the NF2 subset, the main finding we have was they often harbor very complex, copy number alterations. Interestingly, they can also, aside from 22 loss, they also can have 3P loss. So 3P loss is not only just in clear cell RCC. And these tumors often have recurrent co-mutations, SID2, BAP1, and other chromatin modified gene mutations. They have the active HIPO pathway. So this is one typical case. They can be centrally located near the medullary region, and they have this multinodular infiltrating growth pattern, and demonstrate higher power, demonstrate more tubular and tubular papillary growth. So this would, from the traditional point of view, will be very difficult to call what it is. Focally, if you have a biopsy, you could easily call it collecting duct carcinoma. So using the more current strategy, there are not any of those, and then we see the complex copy number changes and the NF2. And so in this particular case, you can see I highlight some areas that shows, different like 2-cell populations and hyalinized material. So these are the ones that has been recently called biphasic hyalinizing psammomatous renal cell carcinoma. But this is just one type of morphology. You can see with NF2, there's some other examples. They can have non-specific tubular infiltrating growth, mixed papillary and sarcomatoid features. And interestingly, when you look at the prevalence of NF2 mutations across the board in RCC subtypes, you can see unclassified is really high, 15 to 20%. Papillary is dropped to less than 5%. It's very rare in clear cell. But interestingly, in some of the other subtypes, you can see this consistent 15 to 20% frequency in FH is one of the most frequent commutations, and in sarcomatoid and in mucinous tubular and spindle cell carcinoma. And for the mTOR subset, now we have known from a more recent study that they include some of the newly identified entities, eosinophilic solid and cystic carcinoma, and eosinophilic vacuolated tumors. However, there are still other tumors that mimic this known or defined new entity, but has higher grade feature, higher nuclear grade, more high-grade aggressive features, and they can present with liver mets a few years later. So those are the sort of vary something for pathologists, so they are always hesitating to classify oncocytic tumors with some unknown features. So in this follow-up study that we presented years ago in meetings, so we expanded the cohort to over 112 cases. So here you see the molecular pathway largely persist, and for this one we already took out the FH group, and you can see the worst behaviors are the NF2 HIPO pathway and all the TP53 DNA damage response pathway mutations. So, so the DDR and chromatin modifying gene mutations often, we consider them later events. So we're trying to looking at what exactly they are because they're lacking other clear-cut drivers. So we're learning from other cases. So on the right, you can see this is the MAT mutated, more classic papillary tumors, but gain a lot of aggressive features we don't traditionally recognize as papillary. So by analogy, and we're hypothesize some of those chromatin modified mutation subset unclassified could represent a highly aggressive papillary. So for treatment, this just lists the unclassified actually show very similar patterns with some of the papillary or collecting duct carcinoma. I think one major challenge we have is how we define the collecting duct and separate it from the unclassified, given the very different treatment modality. So in conclusion, unclassified tumors are high-grade tumors with papillary or poorly differentiated features and diagnosis depending on exclusion. And genomic profiling has identified novel entities and point out them into slightly a few molecular subsites. And as a pathology, I'm hoping the advanced classification in those gray zone tumors can benefit from integration from a treatment data. And what are the tumors actually responding to chemotherapy per se, if there are any? Thank you.
Great.
Thank you. Wonderful talk. All right, great. Well, thank you to all of our speakers. We're going to move into some question answer. We'll have 10 minutes. Raise your hand. We've got Katie in the room who's got the hot mic.
All right, great.
Good morning.
So Lisa, I have a question. In terms of kits, I noticed that between the primary and metastatic sites, there are some differences, either in terms of expression or localization. Can you comment on that?
Yeah, so the question is about KIT expression in the metastatic sites.
And expression versus localization of.
Yeah, so I think that IHC might be a little bit misleading, but it seems the important thing is that it is on the membrane in the metastatic sites, which is where it needs to be for the success of the antibody drug conjugate. So it's expressed in metastases, and it seems to be on the membrane in the ones that we've looked at. I don't know if Michelle Hirsch would have any additional comments about that.
I would just say that it's a subset. We definitely don't see it as diffuse or as strong, but we definitely see patchy and as a KIT expression in a subset of cases.
On that same topic, may I? So chromophobe, when it metastasizes, it mostly does that when it dedifferentiates, right? Whether it's sarcomatoid or anaplastic or a glandular morphology. And those do tend to lose c-KIT. So your thoughts on that? So if the aggressive forms are going to lose it, any thought on the therapy, the ADC KIT therapy?
Yeah, I think that's the key question. How often do the metastases retain enough KIT expression to potentially respond? We'll know if we get individuals enrolled in the trial. But I'm encouraged by the recent results with HER2 new breast cancer, where even the tiniest amount of HER2 in a breast cancer can still result in a response to the HER2-directed antibody drug conjugate. So you may not need very much KIT, and it may not need to be expressed in every cell, especially if you're using an antibody drug conjugate that can be released and reach adjacent cells. So I think it's a very important point, of course, and you have a lot of data and expertise in this, but I'm still optimistic that this clinical trial might give us a really important signal in chromophobe.
Lisa, just to piggyback on that, do we think with the antibody drug conjugate, the payload is chemo, do we think that that cell is going to be sensitive to chemo?
Yeah, so we also thought about this, whether the chromophobes would be responsive. Michelle El-Shaweri, who's here, did try a variety of different payloads in the cell lines that we have available, and they do seem to be responsive. So we'll see. I think that's another unknown that we could best answer that question if those of you in the room who have patients could enroll them in these trials, in the trial.
Naomi?
Hi, so this one is for both the last speaker and Srini. If you had a patient that had an NF2 mutation, then what would be your, just thinking out loud, go-to treatment? Which pathway would you, which agent would you think about?
So, I don't treat patients, but in our experience, I think we have seen a combination of IO with TKI, particularly the trial that Dr. Jolie had conducted at our institution that showed quite encouraging response in a subset of patients. But unfortunately, I feel like in the long term, a lot of those patients still progressed. So I think it's more, to me, I still feel NF2 is potentially an early event that happening. My hypothesis more the distal nephron arising group of tumors that have higher incidence. So it creates maybe a more genomic instability in that setting. So I'm very curious to see whether they actually can demonstrate something more sensitive to chemo agent. But so far, we haven't seen that. We're not really calling anything collecting that these days. Most of the early cases turn out to be FH or some other particular types. So, I think there, hopefully there will be some way to stratify that group better and then test in the preclinical model for certain agents.
Yeah, I would also add, just based on the preclinical data of potential activation of YAP-TAZ, if there are trial options for TEAD inhibitors, which are now being developed, that could be something that could be considered. Maybe.
Yeah, it's a great question, and I think there have been probably dozens of emails just within our internal group at Dana-Farber. I completely agree with Dr. Chen that Cabo-Nivo would probably be our first-line regimen, and then we are investigating phase 1 studies of the YAP-TEAD inhibitors with targeting the Hippo pathway. Michelle, do you have additional comments?
Yeah, I was just going to say, and Yingbei basically also alluded to this by saying some have secondary hits, is that I think it's also important to determine, if possible, whether the NF2, to determine whether the NF2 is the primary driver or a secondary hit, because we're definitely seeing that, as Yingbei said, that there are multiple cases. Some just have the NF2 mutation. Others can have a SMARCB1 deficiency, and NF2, or FH and NF2. We've seen clear cell with NF2. So in those cases, we as pathologists do our best. I know certainly when I have an NF2 mutated renal cell by immunohistochemistry, I still do the other biomarkers to see. I've had cases where you have FH loss and NF, or Merlin is our antibody we use, so you see loss of both staining of both biomarkers, indicating that there's 2 hits. And in those cases, we believe the NF2 is probably the secondary hit, and if nothing else is there, that it's the driver. But it may be that some of these respond differently if it has a primary driver. If it's an FH with an NF2 or a SMARCB1 deficient with an NF2 versus a pure just NF2 mutated. And looking at those separately, the cohorts are obviously not huge, but to sort of look at those separately, I think also matters.
I have maybe a more broad question for the panel. Obviously, biopsies are important, genomics are important. In clinical practice, are we rebiopsying our patients? Are we getting ctDNA? Are we looking more comprehensively? Do we do this at the beginning when we have one of these non-clear cell unclassified subtypes? How often should we be looking? And if you borrow from lung cancer, they're doing ctDNA at multiple time points along the patient's journey. So is this something we should be looking at more comprehensively as patients progress through therapy?
Yes, biopsy frequently and ctDNA frequently, right? I feel like biopsying, getting information, participating in the consortiums, I think we need to tear down the barriers and be able to, collaborate along this point. I think it's critically important to better understand. The point that Lisa was making, we need to sequence the METs, not just the primaries, to better understand what's actually going on. And so I think that as a field, we need to move in that direction.
I think we'll have to end it here because we have a brief break, and then at 8:15 we start back up. Please, we encourage everyone, this is a smallish enough meeting that you can reach out to all the speakers here and please ask questions. This is where we get a lot of ideas. So